So in my PhD, I've been thinking about the issue of how do we integrate genomic surveillance into our national surveillance strategies in England. And the specific aspect of this work I'm going to be talking about is estimating sample size for a specific group of pathogens. So staying on the theme of bacterial bloodstream infections or bacteremias, I'm going to be thinking about for bloodstream infection-associated E. coli and Klebsiella, how much is enough? And I'm also going to be drawing on data from the Nexus study to answer this question, which is the lovely, definitely non-AI generated logo on the right of this slide. So in England, we already have mandatory surveillance of really severe and antibiotic-resistant infections, like bloodstream infections, in the form of collecting patient data and also laboratory phenotypic data. So why do we need genomic data on top of this? So I want to show you, this is a graph of the incidence of resistant bacteremias in England over the past few years. And I'm going to be focusing on the purple and the dark grey bars, so that's E. coli and Klebsiella pneumoniae, that are responsible for the majority of these infections. So we can see that the incidence is increasing year on year. And from this map of England, we can also see that there's inequalities, not just by geography, but also other factors like age and deprivation. And the thing is, we're not really sure why this is. So if we had access to systematically collected genomic data, we would maybe be able to tell which, if any, pathogen genomic factors might be responsible, such as are there specific bacterial lineages, plasmids, or AMR genes that are successful in spreading and causing this increase. And there are many other uses of this data. So, for example, we could use it for vaccine development and monitoring, and these have been really nicely reviewed and mapped to the National Action Plan outcomes in a recent review. So why do sample size estimation? Well, we don't really have the resources to sequence tens of thousands of isolates every year. And the thing is, we might not need to get a clear enough picture of the genomic epidemiology. So I've been really lucky to be involved with this study called the Nexus study, which was a 6-month-long national genomic survey across 10 hospital sites in England, shown on this map. And in this, we estimate to have captured about 10% of all bloodstream infections caused by E. coli and Klebsiella species. And so we've collected about 1,500 E. coli and just under 400 Klebsiella. And for this, we have linked long-read nanopore genomic data with patient data as well as phenotypic data. And so this is a really valuable data set for investigating questions, such as the one we have today. So what's the minimum number of isolates we need to capture a representative proportion of the genomic diversity? So sample size calculations, easy enough, right? Some of us might be having flashbacks to our Stats 101 lectures at this point. But the challenging part about this is measuring diversity in a way that's meaningful for public health. So what I mean by this, let's have a look at the histograms from the Nexus data. So green E. coli and orange Klebsiella. And we can see that they all have a similar shape. They're very positively skewed. So for the genomic units that we mostly look at, so MLSTs, fast-back clusters, which are just different ways of partitioning the population, plasmid subcommunities as clustered by Pling, or AMR genes as annotated by AMR Finder Plus, we can see that most types, so most MLSTs, will be very rare. They occur maybe once or twice, and hopefully this is demonstrated by the schematic below, and there are very few of the types that are common. So now if we counted only the unique MLSTs, we'd be giving equal weight to the rare singletons as well as the common types, whereas in reality we probably care more about the types that commonly cause disease. So my solution to this is using a metric called sampling coverage or sample coverage, which is defined as the proportion of the population that is covered that belongs to an MLST that you've already observed in your sample. And this is beneficial because it conveys this idea of diminishing returns. So this flattening curve, where if you already have a high sampling coverage, you need to increase your sample size by a lot more to get that extra little bit because you're sampling rare species. We have various estimators for this, mainly stolen from ecology, which all have their own problems. So instead, I want to dip into the COVID-19 literature in sample size estimation, where it uses this power calculation, so a simple deterministic equation. And here we can kind of plot curves of, so to detect an isolate of a specific frequency, say 1%, 10% along the x-axis, what's the sample size I would need at a certain level of confidence. And then I want to combine this with my Nexus data, which is the plot on the right. with a method called Bayesian bootstrapping. And here I can estimate the proportion of my population that belongs to an MLST that occurs at least that frequency. So frequency again on the x-axis and proportion of the population on the y-axis. So if I add these two curves together, that's when I end up with this plot, which is my sampling coverage against my estimated sample size. So on this plot, I've shown my Bayesian bootstrapping estimates using the Nexus data in the green, and the grey there is just my ecological estimators for comparison to show that they have a really high uncertainty and they can underestimate diversity. So if we now apply this to our genomic units that we'd commonly use, so MLST, fast clusters, plasmids, and AMR genes, we get these estimates. So again, green E. coli, orange Klebsiella. And if we want to, say, capture 80% of our population at, say, 95% certainty, this is the estimates we get, shown at the bottom visually. So if we look at MLSTs and plasmids, we need about 1,500 to 1,400 isolates for E. coli. If we look at fast clusters, under half of this, so under 700, and in the case of E. coli, this might actually be better because our really big MLSTs like ST131 are actually split into higher resolution, whereas our rare MLSTs are aggregated together. And if we look at AMR genes, we get a very low estimate of just 24 isolates. However, this is probably more of a reflection of the shortcomings of our AMR annotation databases. I also just wanted to highlight Klebsiella. So if you look at the orange compared to the green, it's a little bit lower for all of the metrics, but not by much. And if we consider that Klebsiella has an incidence that's only about a quarter of that of E. coli, we're still going to have to sample a much higher proportion of our Klebsiella population. So in conclusion, Bayesian bootstrapping might be more appropriate. Our sample size depends on the genomic units, target coverage and confidence intervals that we need, and Klebsiella might have more diversity. And that's me out of time, and so I have lots of people to thank, and I'm happy to take any questions.